Hi everyone, thank you for joining us for today's bioCADDIE external webinar. Today's speaker is Dr. John Novembre. He received his PhD from the University of California at Berkeley. Prior to joining the faculty of the University of Chicago, where he's currently an associate professor in the Department of Human Genetics, he was affiliated with the University of California at Los Angeles and was a National Science Foundation postdoctoral fellow in bioinformatics at the University of Chicago. His articles have appeared in journals such as Science Nature, Nature Genetics, the American Journal of Human Genetics, and Bioinformatics, among others. If everyone could please mute your microphones so that way we don't have any background noise during the presentation. And Dr. Novembre, please feel free to go ahead and get started.
Okay, great, thanks. Thank you for this invitation to speak here, and it's really a pleasure. So let me share my screen here. Okay, can everyone see the slides?
Yes.
Can you confirm? Yeah, okay, great. Excellent. So my topic today is talking about methods for characterizing geographic structure and population genetic variation. This has been a research topic in my group for a long time, and before I begin, I want to put human variation into perspective by showing a few major results in the field. And this is one of my favorite figures from early studies of sequence variation in humans in the context of other primates. So this is a phylogenetic tree constructed based on DNA sequences from 72 human samples, as well as samples of several other primates, with the sequence being drawn from the X chromosome. And what you can see quite strikingly is how closely related all the human samples are here relative to other primates. And that is a reflection of our low genetic diversity, that on average across the genome we have about 1 difference per 1,000 base pairs appearing in any pairwise comparison of human sequences. Human genetic diversity decays as you move out of East Africa, and that's a result of humans spreading out across the globe, and as they spread, subgroups moving to colonize each next region of the globe. And so on the top panel, you see a measure of heterozygosity plotted against geographic distance from East Africa, but using waypoints, so distances that pass through the major waypoints that are thought to have been used by humans as they spread out across the globe. When we look at human genotype frequencies and compare them to allele frequencies, a simple assumption of random mating would predict that the genotype proportions are going to be just the product of the allele frequencies. So in the red dots you see in both of these panels the proportion of big A genotypes, where the big A is here always the minor allele at a variant site. And in the thick lines, you see the expectation from Hardy-Weinberg. So if we focus just on the red line, that's the p-squared of Hardy-Weinberg. And you can see that human genetic data actually fits it quite well. In the left side, this is looking at global human genetic variation data. The data come close to the line. They're slightly shifted upward for the homozygous categories and downwards for the heterozygous categories. That deviation is about 10%, and that is measured by a statistic that population geneticists use to measure population deviations, a differentiation called FST. So when we look at genotype frequencies across the whole globe, we get an FST of 10%. When we look within a single geographic region, such as Europe, we get an FST of 0.3%, reflecting a much tighter fit to Hardy-Weinberg proportion. So this is, again, a reflection of how recently humans spread out across the globe. If humans had been existing in separated populations for much longer time periods, we would have, at the global level, a much worse fit. But we're only about 10% off in terms of what you would predict from random mating assumptions. assumptions. And so we really do have this kind of shallow population structure. That said, when we collect large amounts of data, we can use techniques such as principal components analysis and other tools from multivariate statistics and computational statistics to see quite fine-scale structures. So this is a result from a paper we published in 2008, in which, through a collaborative effort led by Matt Nelson and with the analysis team led by Carlos Bustamante, we analyzed about 1,400 European individuals over the Affymetrix 500K SNP data, and when we plotted a PCA for these individuals, we got the following figure that you see there. Each two-letter code is a single individual, and then the large dots represent the median position of all the individuals who have that same two-letter code. A legend for the codes is shown in the upper right, and the two-letter code for each individual is assigned based on their reporting of their grandparental ancestry. So, and we are only showing individuals whose grandparental ancestry information has no conflict, so that all of the information they reported pointed to a single location, such as Spain or Italy or any other location. And what is immediately striking about this PCA is that you can see that the genetic structure mirrors the geographic structure, and that the level of resolution that one can distinguish subpopulations is very fine. As you can see from the graph there. In the context of the studies of human genetic variation and the kinds of results that I was just showing about how close human populations are to approximating random mating populations to having only recently diverged as they spread out from Africa, this result was really striking and impressive at the time that we got it. The result largely arises because of the large numbers of loci that we can look at. So with just 100 loci, you would not be able to detect the structure in any meaningful way. When you get to around 10,000 loci, it becomes more clear, and around 100,000 loci, the basic structure is there. What's important to note is that the proportion of variation explained by these top PCs is relatively small. We are teasing apart very fine scale structure. Okay. And when one scans along the genome looking for places that depart from the overall genomic signature of very low levels of population structure, you will find outliers. So this is a plot from a lab exercise I actually have my students do in the classroom, where they measure departure from Hardy-Weinberg expectation along chromosome 15. And what they'll see is a big spike there in the middle of the graph, and I asked the students to look what genes are underlying that region, and what they'll find is a variance in a gene called SLC45A2, which is a gene that's known to harbor variants that affect skin pigmentation. So skin pigmentation and other, as a generalization, other loci involved in exterior morphological phenotypes like hair color and eye color as well, show much higher levels of differentiation than the background in the human genome. And that's partly because the local selective pressures have favored the rapid change in allele frequencies at these regions of the genome. So whereas in the background there hasn't been enough time, just due to chance, for allele frequencies to change very much amongst human populations, at specific loci where selection has favored rapid change in allele frequencies, you see these higher levels of differentiation. These are, as I said, relatively rare, and I actually think it's fairly profound that the features that humans see in one another in terms of at first glance, in terms of skin pigmentation, hair color, eye color, are actually outliers in the genome and don't reflect kind of background levels of genetic differentiation. So today, though, after giving that background, I want to talk about some more specific methods that we've been developing for studying population structure, and some of these published, some of these under development, and I'm happy to get feedback and from this community and this audience. So first I'll talk about going kind of beyond genes mirror geography in that PCA plot, and so we're trying to look at and develop methods that help us understand inhomogeneity in isolation by distance patterns. So I'll explain that more in just a second. And then I'll describe a browser that we've developed for looking at the geography of genetic variants, and then some new work that we're doing on developing variant level summaries of geographic distribution. So the challenge is that for all of us, we're dealing with large-scale data. In my case, it's large-scale single nucleotide variant data, where we have genotypes for each individual, and so you might imagine the data as a large table with each row being an individual, each column being a single nucleotide variant, and we'll have on the order of thousands of individuals and hundreds of thousands to millions, or even now with the 1,000 Genome Project, 88 million variants to look at, so a very large number of columns. And I take a lot of inspiration from this nice quote from inside of Mike Eisen, Spellman, Brown, and Bodstein's paper from 1998 when they were first visualizing gene expression data and taking great care as to how they did it, where they said, we need to develop our ability to see the information in the massive tables of quantitative measurements that these approaches produce, right? This is not just true for gene expression data, it's true for many other forms of data, including population genetic data. To that effect, population geneticists have developed various tools to visualize population structure. And so these are three major categories of them. One is, on the top, is admixture proportion inference. And the idea here is essentially that you're clustering individuals, but you're allowing partial membership into each of the different clusters. So what you're seeing in this plot is a collection of individuals from populations across the globe with labels along, population level labels along the bottom axis and regional levels along the top. And each vertical bar here represents an individual, and the relative amount of color in each in that bar represents their membership in one of several clusters detected in this data set. So you can see regions of the globe where, like large geographical regions, oftentimes have a single cluster that dominates. So the cluster represented by the color orange in East Asia, blue in Central South Asia, green in Europe, and so we can see that there's a kind of large-scale global geographic structure appearing in this data set, and then oftentimes the populations that are near the edges of any geographic region have levels of mixture with clusters that are dominant in neighboring areas. So this is one particular lens to view human genetic variation through. And another lens is to use factor models or ordination methods like principal components analysis. And as an example, the PCA of Europe that I just showed earlier is one version of that. It's thought that these methods may be preferable for when the genetic data doesn't have such clear clusters, when it's more of a geographic continuum. So that's why, when we move into an area that is finer scale, like a continental region, it may be more appropriate to use methods such as these rather than using methods that assume that there are differentiated clusters in the data. A third category of methods are tree-based models. So this is an output from TreeMix, a piece of software from Joe Pickrell and Jonathan Pritchard, where a tree, a branching tree is used to model the history and relationships between populations, and then edges are added on top of that tree to model any residuals that exist from the tree model that might arise from recent migration between pairs of populations. So each of these processes have their advantages and disadvantages. A lot of my interest is in finer scale geographic structure, looking within geographic continental regions. So I'm going to kind of continue the discussion by focusing on PCA, some of its drawbacks, and some ways that we're trying to provide new methods. So in particular, kind of building motivation for this by stemming right out of that map of Europe result is that when we presented this result, we'd often get the question of whether certain gaps in the map should be interpreted, whether they're real and whether they should be interpreted as regions of accelerated differentiation, somehow barriers to gene flow. So with people asking, for instance, is the seemingly lower density of points that one finds between France and Spain a reflection of perhaps the Pyrenees, or can one see the signature of the English Channel? And I've always been hesitant to try to interpret the patterns this way because of some of the pitfalls of PCA. One of them being that, as we go forward, that if you have clustered sampling in a PCA plot, it will induce clusters in the PCA even if the underlying migration pattern is uniform. So if we can focus on sort of the left side of this figure for the moment, there are what is shown is a sampling scheme where the intensity of the dots represents how many samples were taken. Grid of populations where individuals each generation move to just a neighboring population, and then in the upper panel, in the upper row there, you can see two scenarios for migration rates. One in which the migration rates are all uniform, and that's represented with the white colors across the grid, versus where there is a reduction of gene flow north-south, and that's reflected in the brown color in the middle of that lattice, surrounded by blue colors on the edges. And so if we run PCA on data from the uniform migration case with this sampling scheme that's biased towards sampling the edges of this range with a gap in the middle, the PCA will show a population with, will show clusters. And if there is a barrier, it will also show clusters. So, seeing gaps in the distribution of points in a PCA plot doesn't necessarily give a clear indication as to whether migration rates have been uniform or whether there are barriers. It can oftentimes be as much a reflection of just the sampling scheme. So a more ideal method would actually try to adjust for this uneven sampling. It would use the geographical information that is available for the samples to help do this correction, and yet still provide insight to the genetic relationships amongst the samples. And if such a method could be developed, it'd have a broad set of applications across species, because there are many different groups that are interested in using genetic data to understand what are the major landscape features and geographical features that are structuring the genetic variation in their data set. So with Matthew Stevens, a PhD student in statistics here at the University of Chicago, Desi Flava-Petkova, we've developed this approach and for tackling this problem. And the core idea is to take a set of sample points and with genetic data and infer what you see on the right side of this figure, which is a map of relative migration rates, where here blue means elevated migration rates and brown means a lower migration rates. And in this test example that we have first applied the method to, we're using forest and savanna elephants. So these are elephants that occupy different habitats, and there is not a lot of gene flow across that habitat boundary. And when we run this agnostic method that's only using the genetic data and the geographic locations, it's able to infer that there is reduced gene flow along that habitat boundary. And of course, that's quite a complicated feature to be inferring, in that it's not a single linear barrier gene flow. It has an irregular shape. So this map also shows that high levels of gene flow along the east side of Africa and connecting the various savanna elephants, which is a reflection of how there is a much higher genetic similarity amongst these elephants, and likely because of more easy movement and gene flow in a savanna habitat than in the forest context. So in terms of how this method works in more detail, the way that we construct these maps is to first imagine a grid of populations that exist across space and that are exchanging migrants with one another each generation, and that if we parameterize this model with migration rates and beam sizes, then we can calculate expected pairwise coalescent times. So these are the times back to the common ancestor of individual genes that would be sampled from each of any pair of locations in this habitat. And so when you have expected times to a common ancestor, that's going to be proportional to the expected levels of genetic dissimilarity they're going to be seen. And so that relationship is shown in the bottom equation there of our expected dissimilarity given the collection of coalescent times observed across the grid. And so we can then essentially do a search over the space of migration rates and beam sizes to find a set of those parameter values that minimizes the distance between our observed genetic dissimilarity and expected dissimilarity. And we do this more formally in a Bayesian framework using Markov chain Monte Carlo to obtain these smooth maps where, as I mentioned, like 0 white means average migration rates are taking place. Blue or greater than 0 is above average, and less than 0 brown is below average. The one small comment is that under the hood with this method, we are using some approximations that are based on an idea called circuit theory resistance distances. So the idea, this is a figure from work by Brad McRae, and he kind of took the lead in this resistance distance approximation and applying it in a population genetics context. The idea is that if you're trying to model the genetic distance between 2 points in a habitat, so here are these 2 blue points, these examples for wolverines, that's a critter in the lower left of each of these panels, and the gray area is their habitat. If you were to use a straight line distance between 2 points, that would result in a relatively poor fit. One needs to consider that the habitat shape and migration rates, and so, but once one considers the habitat shape and migration rates, you could try to choose a least cost path that's shown in panel B, and you would get an improved fit over using just straight line distances. But But ultimately, what determines patterns of genetic diversity is not the least cost path, but the average path. You really need to essentially average over all possible paths, and the mathematics that underlie resistance between 2 points in a larger circuit board of resistors has a characteristic of averaging over all possible paths. And that's the same kind of math that's necessary to consider how ancestors of any two genes move through space and traveling backwards in time to find their common ancestor. So these resistance distances work well at predicting genetic distance, and because they are approximating what would be found with exact calculations for expected pairwise coalescent times. And so we actually use these techniques in the Eames machinery. And that's important in terms of accurately modeling the relationship between genetics and geography. And so when we do this, here's a slide now building on the previous slide that I showed with PCA, and where you can see how with PCA, with clustered sampling, you would get clusters regardless of the underlying migration pattern. With Eames, the results are represented just by a surface with colors. And so in the case where there's uniform migration, you can see that the inferred Eames map is actually a flat map, as one would like. And with when there's a barrier, you see the barrier appearing as it should. So this is encouraging for how Eames is working, and we've now been trying to apply it to different parts of the globe. This is with my postdoc, Ben Peter. He's been gathering large sets of more recently published data and merging them, and we've been applying them. And so, when we have that genes mirror geography result where everything looks so cleanly geographical, one might think that Eames will infer a flat map with no local changes in the effective migration rate, which is to say migration rates are just basically constant across space. But we can ask, is there more to the story, and then are there truly wrinkles in the space? As I was saying, in the analogy that genes mirror geography in Europe, is the mirror warped in any sense? And we can ask, will it provide a better representation than PCA, or what kind of advantages will it have over it? And so this is a result of applying Eames to European data. In a first round of the analysis, we see that there are several outliers that do not fit well in the Eames model, and so those are the Sami populations from the high Arctic, Sicilian populations, Latvian populations, and samples of Croats from Bosnia rather than Croats from Croatia. So those outliers aside, the map is shown here, and some of the features that you see are, for instance, low levels of gene flow around the Pyrenees, around Sardinia, which is not unexpected given Sardinia is a population isolate. The Adriatic comes in as a barrier. You do see a lower level of gene flow around the English Channel, in the Baltic, and while water features tend to be areas of lower gene flow, you see that the North Sea is relatively close to zero. It's not as strong of a area of low gene flow as, for instance, the Adriatic, English Channel, or the Baltic are, and that may represent connections between Scandinavia and the British Isles. Also, you can clearly see this sort of high migration area between Scandinavia and Iceland, which of course is not unexpected. So this tool, we think, is representing a lot of very subtle patterns in population structure that once you include the geographic information in this explicit model, that you can see quite clearly and gain a lot more subtle understanding than simply seeing the broad scale patterns of genes mirroring geography at a coarse scale like we did in the PCA. And just as a reflection of this, if we plot genetic similarity versus geographic distance, straight line distance, we get relatively poor R-squareds is like 0.26. If we use the fitted dissimilarities provided by this migration surface, we get R-squareds of 0.84. If we compare that to the dissimilarity that we get from just looking at the distance between points in the PC1, PC2 space, the R-squareds are about 0.68. And extrapolating that, so looking at distances in 3, 4, 5, 6, 7, 8-dimensional PC space, they still do not result in as good of an R-squared fit as using the single EAM surface. So that is to say that we think these maps, while they take some adjustment to learning how to read them, are a two-dimensional representation that actually is encoding a lot of a structure that exists across multiple PCs and would require looking at several different biplots to try to uncover otherwise. So we have a couple other examples. In the interest of time, I'm just going to move right on, though, and talk about future directions. A future direction for us is actually to use new metrics of genetic dissimilarity. So for EAMS as it is, we're using a metric that's based on single allelic patterns, like variant patterns. We don't consider the haplotypic context of the variants. And in our recent work, we are looking at long shared haplotypes between individuals, which are reflective of very recent patterns of genetic similarity and ancestry. And so because they reflect recent ancestry, they're very sensitive to what the recent patterns of gene flow are. So if we look on the right side of this figure, the idea is that we're doing a simulation where there is a barrier to gene flow greater than 20 generations ago that then disappears. And EAMS, which is only using single variant statistics and inherently, and especially it's based on sort of average pairwise coalescent times, it inherently averages over long time periods, and its inference reflects the long-term persistence of this barrier. But when we use this new method, which we're calling MAPS, and look at long shared haplotypes of different sizes, in the longest segments at the bottom of the figure here, we infer the flat map that is representing what is happening today. And if we use longer segments, like those from 2 to 6 centimorgans, we can infer the barrier that you see in the middle there. Okay, so we are kind of enjoying these methods and finding a lot of utility. And our dream for these is to actually someday provide maps that are time slices, slices through time that show how migration has been changing and population sizes have been changing, to really reconstruct a history of population movement. A challenge, though, that I find in developing these kinds of methods and using other methods for visualizing population structure is that none of these methods convey absolute levels of differentiation very clearly. So one has to look at affiliated statistics with each of these visualizations. So in structure, you need to look at the levels of FST that exist between the inferred clusters in PCA, the proportions of variance explained. In EAMS, the average migration levels. In tree mix, you have to have an understanding of the drift parameter. So none of them, even with those numbers, actually, I'd argue that none of them really help convey to a user what are typical allele frequency patterns. That is, if I were to grab a single nucleotide variant from one of these data sets, what are its allele frequencies likely to look like? So we've been motivated by that to develop approaches that allow one to look more quickly at what variant level data is like. It also, these tools have utility for researchers who are just interested in a specific variant. But first, in terms of conveying, what is a more typical allele frequency pattern, one thing that I would do often in my early presentations on that PopRes gene smear geography result was to include this slide, which had frequencies for the minor allele. That's the rarer of the two alleles at a single nucleotide polymorphism site, and its frequency plotted across different regions of Europe for just 25 random SNPs. So while these data sets are large and one can't hope to do this in a useful way for the hundreds of thousands of SNPs that exist, one can subsample to a manageable size and get a sense of some of the average behavior. So what one sees here is that allele frequencies hardly vary across geographic regions of Europe. The fluctuations are on the scale of only a few percent. And one has to bear in mind that some of the fluctuations that we see with our naked eye here are fluctuations due to sampling variation. That the underlying population frequencies actually probably vary less than what is seen on the graph, which has the standard error of having finite sample size. So this helps one see that despite the fact that we can build these PCAs of Europe and using genome-wide data have a sense of whether someone comes from southern or eastern or western Europe, that one can at any SNP that information is not very predictive of frequency levels at any given SNP. So the spirit of tackling big data by visualizing random subsamples is something that we find very valuable. And so we've been trying to develop these better tools to show how allele frequencies vary across space. And we're trying to extend upon some work that we had done, and I had specifically done in my postdoc with Joe Pickrell and as part of a project on analyzing data on the Human Genome Diversity Project with Jonathan Pritchard's lab. Joe and I developed a browser and scripts for plotting the geographic distribution of any variant that was on the Illumina array that was genotyped in the HEDP. And this is something that we had up in the HEDP selection browser. We still have it up, and that one can resource through the UCSD genome browser. And provides these nice maps in a PDF format or other formats, and researchers have found them useful, and we see them in published papers because they make immediately a nice figure that conveys a key aspect of a variant's biology, which is its geography. So this resource has become sort of less relevant given that there are now genome-wide data sets. And so what we've been doing is trying to develop a kind of new resource that accesses the newest data, like the 1,000 genomes data. And that resource is the Geography of Genetic Variants browser that we've developed. The main lead has been Joe Marcus, who was a programmer with me and now a PhD student. It's a D3-based browser that accesses a backend that is a REST API that allows you to pull frequency data from several of these data sets in JSON format, and we think that's its own contribution for people to quickly get access to frequency data without having to do as much data wrangling as immediately downloading many of these data sets directly. And what I'm showing here is a screenshot from the browser, what one can see is, here I'm using a pointer, a drop-down box for different data sets, a search window based on RSID or chromosome position. Crucially, we have this random button, which I'll demonstrate in just a minute, which allows you to see random variants in the data set. And as I was mentioning, I think that's really helpful in conveying some of the average properties of a data set relatively quickly. Then we have a view of the sequence context of where that SNP is, what genes are present, and then sort of mini genome browser below. And we have the ability to export as PDFs so that people can use these in figures. Now, there are a couple visualization challenges that we had to face in scaling up to these new data sets that have much larger sample sizes than the HEDP did. And so one of them is that we use this frequency scale here, where, as I'll show you in just a second, we actually change how the frequency scale is measured depending on the variant. And then we also have this approach for showing sample size and reflecting uncertainty. So the idea of this frequency scale is that sometimes the variant is extremely rare. So in the example in the map here, this is a variant that's just below 0.5% in frequency. And if we were to use this pie chart formulation raw, it would be a tiny sliver that would be difficult to actually extract a numeric value out of quickly by eye. So we scale down the proportion that we're measuring out of. So rather than measuring a proportion out of 1, as we do for a common variant, like in the previous slide, we measure out of 0.01, so that in the example there, something that a pie chart that's 25% full is actually 0.25%. And once you get used to this color scheme or to viewing, for those who are colorblind, the number in the legend there, one can quickly read off these frequencies, even though they're varying by orders of magnitude. And so yeah, we do have this system where we move between different colors and demarcating different scales. And essentially this is a kind of a scientific notation with pie charts, which allows us to represent frequencies across different orders of magnitude. It may not be the ultimate solution, but it's something we find works quite well and solves this problem. And then a challenge with representing frequency data in this context is representing uncertainty in the data. So that these frequencies are estimated from finite samples and can have substantial standard errors. And so a trick we use for that is to make points increasingly transparent in proportion to how small the sample sizes are. Okay, and so here what I have is a kind of zoomed in view of the browser and a little animation that is from hitting that random button on the browser. I suppose since we are on a webinar and I'm screencasting, I can take the leap here and actually show you the browser itself, and we can hit the random button and do this live, which might be more fun. So here's the browser here, and one more point is that you can access any particular variant via URL query, which is of course useful for people linking in to drawing a specific map. And if we hit the random button here, so we see, this is avoid overlap, that this is a variant at about 0.5% frequency in Africa. Hit it again. Okay, this is a common variant. These are frequencies of about 47%, 26%, something that existed in Africa, spread out across the globe, a common variant. Okay. Then here's a rare variant in East Asia, about 0.5% frequency, rare variant in the Ceph, Utah population. Here's one that's rare again, 0.5%, and about 1% in Southern Europe. This is about 1% in East Asia, localized to East Asia. So the variant that's in Africa and in African descendant individuals from the Caribbean. And so what we see as we click this through are lots of rare variants that are geographically localized, right? Occasionally variants these are a little more common. These are about 8% in Africa, found in African Americans and Afro-Caribbean individuals. This is the kind of variant that, a simplistic interpretation is that it existed in Africa, and then as human populations moved out of Africa, it did not spread, and by admixture made it to the Americas. So we kind of click through, and we get a sense of what human genetic variation data looks like with lots of rare variants. and much less frequently common variants. And those common variants, you can see, tend to be widespread. Okay, now that's a little hint of what human genetic variation looks like. I actually would love to see this kind of visualization feature included on many more genomics browsers to just give us it's easy to begin getting a feel for the data by going through that exercise. At the same time, of course, random sampling is always prone to error and misrepresentation, and so I've been trying to develop an approach for getting a genome-wide summary of the geographic patterns observed. So how can I provide a more clear representation of how often we see those rare variant patterns versus those common variant patterns? And to do this is a challenge in that we have to take this geographic map, which is essentially a vector of 26 numbers of frequencies, and then somehow clump them into patterns that are meaningful to us as humans, right? So, for instance, the first map that I'm showing there is a pattern where a variant is rare in Europe, right? I rarely am a human biologist rarely concerned about exactly which population it's rare. It's just that it's rare in a broad geographic region. It's sort of useful to know. And then the map below is that it's common globally, that the frequencies are about the same across the whole globe. We'll call it common globally. This problem is akin to problems of doing clustering of gene expression patterns and across different experimental conditions or across time series. And for the moment, we're actually taking a very simple-minded approach, that is to make a hard categorization of whether the frequencies are basically zero in the observed data, so that we call a variant undetected or absent, so that's with the letter U, that the variant frequency is rare, meaning that it's less than 5%, or C, common, which is greater than 5%. These are frequency thresholds that human geneticists and the medical genetics community use in practice in talking about variants, so it becomes a kind of natural way to do a threshold. And then we use the 1,000 genome superpopulation. So these are rough continental or subcontinental scale regions that the 1,000 Genomes Project groups the populations into. And so you take 5 superpopulations by 3 frequency levels, and you have this 5-digit code where there are 244 possible codes, and on the right side there you see examples of how the map of 26 values gets boiled down to the simple code. And we actually think providing these to users in the human genomics community will be useful, because they can have in tables representations of not just the p-value for their variant, but this little 5-digit code that would immediately communicate its geographic distribution in a compact way. Now, once we have these, we can go through the 88 million variants in the 1,000 genomes data and ask what categories would be the most common genome-wide, and ask how many of the 243 possible categories actually explain some large fraction of the variants. And so this is our result here. What we're showing is the proportion of variants that fall into each of the different geographic categories. And what we see is the most abundant category, the one on the bottom here. Sorry, I'm trying to get my mouse cursor back, but can't seem to. So the RUUUU, which is rare in Africa, absent everywhere else. Then rare in East Asia is the next most common, with 11 million variants. Then 10 million that are rare in South Asia. Then rare in Europe. Then rare in the Americas. Then rare in Africa and the Americas, and that's partly because of the African descendant populations in the Americas. And then when variants are common, they tend to be common globally. This was something that we were seeing in our random sampling of different variant frequencies. Then we see a category that's common in Africa and absent everywhere else. These are the variants that are probably lost during the bottleneck out of Africa. Then rare variants that are shared just between typically 2 or 3 geographic regions that are neighboring each other typically, or connected via the recent migrations into the Americas. And then other patterns are much less common. And so it's quite striking that actually, despite the 88 million variants that exist in the 1,000 genomes, they actually fall into a relatively small number of categories. And that a large number of these categories, actually, these ones where it's rare in one part of the world and absent everywhere else, these are variants that essentially everyone in the world has the same genetic material, except for a few individuals in one pocket of the globe. So we find this kind of compelling as a compact summary of the structure in the 1,000 genomes data that not only conveys a bit about how populations are related to one another, but directly conveys about allele frequency patterns. And it's a tool that we can use in different settings. So, and I'll show examples of that. But first, let me make a comment about this relative abundance of rare variants that are geographically localized and common variants that are geographically widespread. So, human populations, as I mentioned at the beginning of the talk, are only very recently distributed across the globe. And so variants that were common in our ancestral population in Africa will have not had enough time to actually be lost in any one population. And so if it was common in the ancestral population, it's probably common across the whole globe. And so we see things that are common tend to be globally shared. Meanwhile, once we spread out across the globe, there has been enough time for new mutations to arise, but not for them to spread to any reasonably high frequency. So most new variants are still rare and localized. And so that's why we see this set of patterns with a single R and the rest U's, and then the next most abundant being the C's, where it's C shared everywhere. So if human populations had been diverged for a much longer period of time, you might see lots of cases where there's a common variant localized to a single geographic region, but there just simply hasn't been the time for that to occur. So this pattern is characteristic of human demography and tells us a bit about our history. I've run simulations where you assume this demography and you actually don't. I should make one more point. If population sizes have stayed constant, you would not see such a large abundance of rare variants. So you need population sizes to be growing, which is a feature we understand about human history, to see this large abundance of rare variants. So that we've spread out across the globe quickly, that we and then hence have grown in numbers, that we have such a large number of shared common variants and a large number of rare variants that are geographically localized. Okay, and like I said, that's a signature of our spread across the planet. This tool then can be used for looking at different types of variant lists. So when we apply the same technique to visualize the geographic patterns of variants that are on different array products, we see how they are greatly enriched for common variants that are spread across the globe. This is a well-known feature of ascertainment bias, but we can see more subtlety here in how some of the early array products are relatively enriched for variants that are at higher frequencies in European and South Asian ancestries, and then as we move towards more modern or recently developed arrays like the mega array, the multi-ethnic, the ME is for multi-ethnic genomic analysis array, that there's much better representation of variants that are rare in African ancestries. But at the same time, you can also, as you scan that fourth column of the East Asian ancestry, you see lots of U, that white color, representing that these arrays still relatively do not capture strongly East Asian ancestry variants. So now when we we can also apply this to looking at personal variants. These are the variants that appear in a single sequence individual's VCF file if we were to sequence them. So for instance, if an individual is GT heterozygote when the reference is a G, and apply the same approach, we see that for single individuals, the variants that you or I carry, they tend to be common variants. So while most variants in the 1,000 genomes are rare, it's rare that any of us will have them. So most of the variants that we carry are actually common and geographically widespread. So most of the ways that we differ to the reference or differ amongst each other are variants that are globally shared. And this has been a core realization of human genetic variation for a long time, but this is a new way of visualizing it. So in the interest of time, I'll skip over one more application and get to caveats. So we're using the 1,000 genomes data, which is a low coverage study design, so it may misrepresent some of the geographic patterns. These category assignments have these hard thresholds, which may cause issues with smaller sample sizes. We are actually uncertain about whether variant frequency is rare or common, or rare versus absent. And we actually have a probabilistic machinery under this that allows us to take that into account. And when we do, the global picture only shifts slightly. So we're finding this new method to be useful and a new way of visualizing allele frequency data. What's ongoing is doing things that are a bit more sophisticated, where we use the full 26 population data and some careful clustering methods to help reveal some of these patterns. So just to zoom us back out, I began the talk by talking about this Eames method, and one of the main punchlines of that was that major topographical and marine features regularly are affecting patterns of variation in humans, and that this method provides a good alternative to PCA-based methods where it anchors the samples with their geographic position. We still have a lot of work to do in terms of addressing caveats in terms of persistent issues like uncertainty in sample locations, dealing with outliers in the model, making flexibility to the grid sampling that we use, and dealing with time-dependent migration. So I'll just skip to my acknowledgments. I want to thank my lab members who've been extremely helpful with all of this work, especially Joe Marcus on the GGB, and Ben Peter on the Eames project, Desi Petkova, and Matthew Stevens for the Eames work, and Matt Nelson and Carlos Bustamante for their leadership in the PopRes project. And of course my funding sources, including the BD2K. So hopefully it'll be just a couple minutes for some discussion and questions. I'd love to hear your feedback.
Are there any questions? Okay. Well, thank you so much, Dr. Novembre. And just remember, everyone, our next webinar will be Thursday, April the 13th. Thank you, everyone.